Hi, my name is Benjamin Escandon Rodriguez, and I'm going to present you my project about the expression of human IL-33 protein in mammalian cells. First, I'm gonna talk a little bit about interleukin-33 or IL-33 protein. It's a member of the IL family of cytokines involved in the production and maturation of Th2 cells and mast cells. It can function as a traditional cytokine and as a nuclear factor that regulates gene transcription. It is also thought to function as an alarmin released following cell necrosis to alerting the immune system to tissue damage or stress. To express the IL-33 protein, we need to design a vector capable of delivering the DNA sequence into the mammalian cells. To carry out this process, I decided to use the pTarget mammalian expression vector system because it allows to clone PCR products with T overhangs. It uses a CMV promoter enhancer that's robust and is capable of a consistent expression. It also uses a CV40 enhancer, allowing the increase of the level of gene production, and it also contains a neomycin marker for the selection of stable transfectants. We can also see here the vector circle map and the sequence reference points. Following the guidelines in the manual, we can construct the PCR product. The vector is already pre-clipped, so we just need to add the PCR product where it says cloned insert. The pTarget T vector shows its promoter and multiple cloning regions. The top strand of the sequences corresponds to the RNA synthesized by T7 RNA polymerase and is the same as the single-strand DNA produced by the vector. The pTarget T vector also contains a modified version of the coding sequence of the alpha peptide of beta-galactosides, here known as LAGC alpha region, that allows blue-white recombinant screening. The codons of the LAGC alpha region within the multiple cloning regions are indicated by the spacing of the nucleotides. First, the pTarget mammalian expression vector system manual says that the vector needs to be prepared by coding the vector with EcoRB and then add the 3' terminal thymidine to both ends in order to have the T overhangs. The manual suggests that we use the pGEM-T vector system 1 for the PCR cloning, but we can also use other systems. The next step is to carry out the ligation. In this step, the PCR product is added and the conditions are shown in the table. The manual suggests to also do a positive control and a vacuum control without the PCR product. T4 DNA ligase included is necessary because it allows the thymidine to exist in the vector. Without it, it will disappear. This process needs to be 3 hours long minimum and is a huge disadvantage to other systems. The final step is to realize the transformation using JM109 competent cells included in the system. There is a possibility to use other cells, but the use of JM109 competent cells is recommended as they are ready for blue and white selections and screening. Using LV plates with ampicillin IPTG and NextGal is necessary in the transformation, and the LV plate should contain 100 microliters of cells. We can validate the process by using the Sanger sequencing with T7 promoter primer or pTarget sequencing primer. Both of these primers are included in the system. Here is the reference of the manual.